Oogenesis is a type of gametogenesis where female gametes, that is ova, are developed within female reproductive organ, that is within ovary. Human oogenesis starts before birth, that is during fetal life. After birth, ovum production starts at adolescence period, that is at the age of 8 to 13 years of female individuals. Ovum production is stopped after 40 to 50 years of age, but ovum production temporarily ceased during pregnancy period and during lactation period of a woman. Oogenesis consists of three phases. 1. Multiplication phase, 2. Growth phase, and number 3. Maturation phase. This is schematic representation of oogenesis. First, multiplication phase. This phase occurs before birth, that is during fetal life at the embryonic age from 15 week to 7 month. Primordial germ cells, which are diploid in nature and contain 2n sets of chromosomes, undergo mitotic division or divide mitotically to produce a couple of million oogonia within each fetal ovary. No more oogonia are developed or formed after birth. So, every female child is born with a fixed number of oogonia. The oogonium then again undergoes mitosis cell division, produces primary oocyte. Primary oocyte is also diploid in nature, contain 2n sets of chromosomes. But here, meiosis cell division also starts, but arrested in diplotene substage of prophase 1 of meiosis 1. So, they are still diploid in nature. So, this is multiplication phase. And occurs before birth or during fetal life. Then birth takes place. Now, growth phase after birth. Each primary oocyte is enveloped by a single layer of follicular epithelial cells and is called primordial follicles. So, primary oocytes are transformed into primordial follicle. Primordial follicle is then transformed into primary follicle. At the time of birth, total number of primary follicles within ovary about 2 lakhs to 20 lakhs. A large number of primary follicles are degenerated from birth to puberty. At puberty, only 40,000 primary follicles are left in each ovary, and this degeneration of primary follicle is called follicular atresia. So, transformation of primary oocyte into primordial follicle, transformation of primordial follicle into primary follicle, and follicular atresia, this portion is called growth phase. Third phase, maturation phase. In this phase, secondary follicle is transformed into Graafian follicle. Primary oocyte within the Graafian follicle completes its first meiotic division and forms two unequal cells. One haploid cell, that is called first polar body, and another large haploid cell which is called secondary oocyte. Immediately after meiosis 1, secondary oocyte initiates meiosis 2, but the process halted at metaphase 2 stage until penetration of sperm takes place. Then ovulation takes place and secondary oocyte is released, which is called oocyte and also haploid in nature. This is oocyte, and this oocyte is surrounded by a layers of cells which is called corona radiata. Now this oocyte enters into the fallopian tube. So now oocyte is within the fallopian tube and it is surrounded by corona radiata. Within fallopian tube, nucleus of the sperm enters into the oocyte, but fusion does not take place. Fusion does not take place, and this is called now ovum. That is, before fusion of these two nuclei and then fusion or fertilization takes place. As a result, zygote is formed. When the sperm penetrates oocyte, meiosis 2 is completed, and as a result, actually, after ovulation within the fallopian tube, secondary polar body or second polar body is created, which is haploid in nature. Sometimes first polar body divides and produces two cells, and later all polar bodies are degenerated and only ovum is left. So there are three phases. One, multiplication phase. 2. Growth phase, and 3. Maturation phase. Hormonal control of oogenesis. Oogenesis is controlled by various hormones. Let us see. Hormonal control of oogenesis. During oogenesis, hypothalamus of the brain secretes gonadotrophin releasing hormone or gonadotrophin releasing factor, GnRH or GnRF. This GnRH induces pituitary gland to produce LH hormone, that is luteinizing hormone, and FSH hormone, that is follicle stimulating hormone. These two hormones play a major role during oogenesis. LH hormone or luteinizing hormone induces ovulation, and follicle stimulating hormone induces growth of Graafian follicle. And under the influence of FSH, meiosis 1 is completed in primary oocyte within Graafian follicle. These two hormones are very sensitive to ovary during puberty, and these two hormones together induce menopause. During middle age, a woman's ovary is less sensitive to LH hormone and FSH hormone. As a result, ovulation is stopped and growth of the Graafian follicle is stopped. As a result, menopause occurs. So, this is the hormonal control of oogenesis. Thank you for watching this video. Stay with the channel and subscribe the channel to get more videos.